So I'm going to do this one as a, this is the actual screen of my laptop, and we're going to do it this way. If you hear that fan in the background, that is my computer trying desperately hard to render these things to YouTube. Yeah, bless it. Boy, yeah, YouTubers, man, I don't know how they do it. Okay, so we're going to go through a series of videos. That's why I'm doing this. Yeah, so let's look at these things. What we're going to talk about is how the Picornavirus actually goes about its translation. And so here's some videos. Let's just have a little browse through them. All right, so we'll start out back here with, there we go.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for.
Okay, and so the reason we're doing this is because picornaviruses take advantage of a loophole in translation, and they break the ribosome so that only they can use it. They are the dirtiest little cheaters. But we got to kind of have a pretty good picture of what a ribosome is and what it does for all this to make sense. So let's try this.
There can be several million ribosomes in a typical eukaryotic cell.
Okay, so the blue thing is a ribosome. The yellow thing is a messenger RNA. These little green things that are flying in from every side are transfer RNAs, and so each one of these has one little amino acid on it, and they're going to get loaded into this machine. They're going to line up with the right three bases down here and the right three bases there, and then the ribosome's only job is to make sure they get a good fit and then take off the amino acid and add it onto the chain it's already building.
These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works.
Let's see, yeah.
The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape.
From back when computer tapes were a
The
thing,
amino
I guess.
acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA.
Yeah, and if you happen to be colorblind, let me circle it. This is the amino acid, and the rest of this big pork chop looking thing is the transfer RNA. So that's what they're talking about.
The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site.
So A for acceptor, so this is where we accept a new tRNA coming in. P for peptidal transfer, so we're going to take that amino acid off and transfer it onto the peptide, so peptidal transfer. And E is the exit site, or you can also call it the ejector seat. It's just where we chuck out the leftover tRNA that we've already taken the amino acid off of. So here's a new tRNA coming in, and there's this little amino acid waiting right up there, and that's what we're going to take off, but we can't take it off until we move it to the P site. And so amino acids, or rather tRNAs, come in at the A site, move to the P site, then get ejected from the E site, and on and on and on and on, just like that.
The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA.
So the anticodon is, so the tRNA is made of RNA, and a messenger RNA is made of RNA, and so they all have the 4 RNA bases. They have A and C and G and U instead of T, because this is RNA. And so if they make a match, so if there's an A on the codon, there should be a U on the anticodon. And if there's a C on the codon, there should be a U on the anticodon. Anticodon is just the part of the tRNA that slots right down in there and gets to make contact with the messenger RNA. And if that part matches, then it gets to move from the A site to the P site. That's all there is to it. Okay,
Next,
so let's
provided
jump
there is
out
a
of
correct
that one.
match, the T.
Yes. Boom. And actually, we'll skip over this one. So I put up here a nice little thing with bacterial translation. It's a little different, but the molecules are just great on here. And what it shows is that to start out translation, this is the small subunit, this brown thing, but it also takes a bunch of other initiation factors. Now, they're different factors, or rather, they have different names in eukaryotes versus prokaryotes, but it's these factors that are going to be the key to our story for how the Picornavirus is able to do this weird and terrible thing. So I'll leave that for you. You can watch it. It is linked over in Blackboard. Boom. On to the next one. Now this one wouldn't have made much sense before maybe, but this is the one that's going to be most like the test question. So what you've got here are some parts. Let's look at these parts. We've got the 40S ribosomal subunit is the small subunit of the ribosome. So this is that thing that normally looks like a hand making sort of with like 3 fingers on it, basically. In real life, but they've just drawn it as a little sphere on here. That's great. Each of these other things is a thing called a eukaryotic initiation factor. So eukaryotic initiation factor 4E binds to the cap. And so a cap is a guanine molecule with a methyl group at the 7 position. That's the N7 position, as in the N7 methyltransferase. And that's the reason why we have N7 methyltransferases, because eukaryotic initiation factor 4E has to bind it, or else there is no translation. So 4E binds the cap. 4G is a bridge between 4E and 4A. 4A is a helicase, so it's going to run along and open up the any secondary structure, any sort of folded bits in the RNA, and make sure that the ribosome can proceed. And then 4B and 3 are both going to connect up to the 40S ribosomal subunit. So these are all going to come together and interact. And there are a couple other parts that interact as well, but these together are going to be the initiation complex. So the important ones are 4E, 4G, 4A, the 40S, and then 3 and 4B, which used to be called 4F, I think, collectively, but are now called 3 and 4B. That's a better way to think of them. All right. So those are the parts of the ribosome. Here, let's zoom this in a little bit. There we go. There we go. Okay. But instead of just a normal messenger RNA with an AUG, that's a start codon, so that's where we're actually going to start translating, the Picornaviruses go one better. So they fold up their entire front end. They have these really long 5', like at the beginning of the genome, untranslated regions, and they fold up into a thing that's, I don't know, kind of like a crazy clover leaf or something. It's called a clover leaf structure, but as you can see, it's just a bunch of little bulges and little stems on there in between the bulges. And it folds up in such a way that its AUG is right down here on a little loop. Now that's going to be important. There are extremely important parts of this thing, and what you've built here is actually a machine made out of RNA. And this is going to wrap around the ribosome, and it's going to make the ribosome feel as if it is bound to a complete complex of EIF4E and EIF4G and EIF4A and EIF3 and EIF4B. So it's going to simulate that sort of experience for the ribosome in terms of the molecular interactions. So there it is in 2 dimensions. And then we've actually solved the structure of a couple of these together. they sort of, they make this sort of structure. Now here, you can see what that structure is for. So it's reaching around, and it's actually making contact in all the binding sites, and it's making contact with the places where normally factor 4G and 4A and 4E would be binding and making contact with the ribosome. And so because we're doing that, now we don't have to have those other components. And it just happens to deliver the RNA, the translation start site, rather, the AUG, into just the right spot on the protein. So, yeah, comparing here, this is the small subunit with some of these factors on here. This is the helicase, I believe, 4A. And so those are bound and being held together by the virus. And so this part of the virus is called an internal ribosome entry site. IRES. Make sure you get that in your notes. I for internal, R for ribosome, E for entry, S for site. And it's named that because when people first discovered these things, they found out that, wow, you can get this to, you can get a ribosome to bind to this site and it'll make a protein no matter where it is in the RNA. Because ribosomes normally would only make the first protein on an RNA and then would stop. Yeah, so yeah, that's how it works. Really, it's more of a direct ribosome binding site. And so you can see it is simulating. The purple thing is holding these pieces together in the same way as they would be held together by factor 4G, which is up here in blue. Yeah, so that's what we're going for. Okay, and once the small subunit is assembled with those initiation factors, then the initiation factors pop off and leave, and bam, we're in just the right place to start translating that Picornavirus. So we're going to go back, and I'm going to do a little drawing of that so that we can really see what's going on there. I think that's going to help. So yeah, I think that's probably a good place to pause this video, and let's go over to the drawing, and then we'll come back and do the rest of this section.